introduction yesterday. I am Raj Singh. I work at UCLA, and I work with a team of people. This is a small list that we have 15 people in my lab who work on different aspects of autoimmune disease or immune system. We collaborate with lots of people. We collaborate with people in endocrinology at Harvard UCLA Medical Center, and KS community knows Ronald Sherlock and Christina Wong quite well. They have studied Klinefelter syndrome for a long time. They've also created Klinefelter syndrome mouse, and they have done all kinds of different studies. And we collaborate with several people around the world. So today in this presentation, in the first few slides, I will talk about why I'm giving this presentation, what is the rationale for the studies that I am doing. And because this crowd may not be, if there are no immune system talks to this crowd, so I'll discuss what is the immune system. Okay. And then I'll discuss a little bit about my data. I won't go into detail. Maybe next meeting when I come, when you already know everything about the immune system, then I can talk about what goes wrong with regards to immune system in people who have Klinefelter syndrome or other XY variations. So this slide is half of the story. Okay. So basically, sex chromosome variations can affect immune response in all kinds of ways. So first, the gene SRY is a master regulator gene in terms of sexual differentiation. If one has SRY gene, one develops testes and becomes a man. You take away SRY gene and this person develops ovaries and becomes a female. And these estrogen and testosterone, they have all kinds of effect when they signal in cells, causing all kinds of changes in immune cells, in addition to all kinds of other cells. Now, the sex chromosome, there are lots of genes on X chromosome, there are few genes on Y chromosome. Okay. And these are called X-linked genes because they are present on X chromosomes. Okay. And these genes talk to or have effect on the regular 22 genes on regular 22 chromosomes. So there's a lot of interaction between them. And the sex hormones can affect immune responses. X-linked gene can affect immune responses. And there's a crosstalk between all kinds of different components. So basically what it tells me is that it will be a difficult job for us or other people who are studying to exactly figure out what is, if there's a phenotype, if there's something abnormal, what is the cause? Is it directly because of this, because of genes on X chromosome, or because of their effect on the 22 autosome genes, or because of their effects on hormones, and how hormones and chromosomes talk to each other. Okay. And this is a small list of genes on X chromosome. And what I'm going to do for the next 70 days, I'm going to discuss the effect and function of each of these genes. Okay? And this is only a partial list of genes. This list of genes is important in, it can cause all kinds of immune changes and play a role in the immune system. So be prepared. Okay. I showed this review article yesterday because this is an interesting review article where it says the X chromosome, in the immune system, X chromosome can make a difference. So in response to various immune challenges, females show immunological advantage. They respond to immune response better. The X chromosome has an important role in this immunological advantage. And I was just talking to one of the parents that in the beginning when we started doing these studies, we thought, yeah, this will be pretty simple. X, Y individuals will be like XX because they have two X chromosomes and the immune responses will be similar, but that is not the case. There are lots of differences. So just X chromosome dose is not enough or not sufficient to explain the differences. However, on one hand, females have advantage that they can fight infection better. Actually, for some reason, if you talk to any basic immunologist, they always do a study in female mice. And everybody, so when I came and started doing research, everybody told me, look, just take female mice, do this experiment. People don't study male mice. Except in the last like 5 or 10 years, people have begun to compare what are the differences between male and female mice. Because female mice mount very strong immune response. However, females are prone to autoimmune diseases. The strongest genetic factor, and this is the slide made by Eric, is being female. So generally speaking, females are 3 to 10 times more likely to have autoimmune disease compared to males. And this is a small list of autoimmune diseases. There are at least 81 different autoimmune diseases that affect different organ systems. And the pink is the prevalence, incidence in females, and the blue is in males. And you can see certain autoimmune diseases are really common in females, 13:1 ratio or something like that. Okay, but not all autoimmune diseases are like one. Some actually are not different. They're similar between men and women. Okay. The question that we have been interested for the last 7 or 8 years is, does sex chromosome genotype play a role in the development of immunity and autoimmunity? Is it just a question of dose, or how much dose the X doses or Y doses, how much is the contribution of the sex chromosome genotype? Okay. And we did begin this study in mice. And these are very interesting mice. And in this mouse, as I told you earlier, if you take away SRY gene from the Y chromosome, now this becomes female. So now you have XX female and XY female. So for us it becomes really interesting because now we can compare immune responses between this mice and this mice. They're gonadally similar. They have similar hormone levels, but they're different between X chromosome genes. Okay. Now the other thing you can do is you can take that SRY gene and make what we call transgenic mice. And in this mouse, we take this SRY gene and put in one of the 22 chromosomes. Now these become male. So now you can compare XX male and XY male. So basically they have male hormones, similar, and differences between the chromosome. Similarly, you can take this mouse and compare between the two, the differences between hormones. You take this mouse, and they have same genes, but difference is in hormone. And you can, these are mice, you can take away their gonads, castrate them, or take away their ovaries in early life, and then you can see the effect of pure chromosome, or keep the hormone, keep the gonads intact, and can see how sex chromosome affects immune response, or whatever you want to study, in the presence of normal hormones or in the absence of hormones. And then basically this is what we are comparing. So we are comparing this versus this and this versus this. Yeah. And this is one of the experiments we did. So here, in this axis is survival from an autoimmune disease called lupus. Okay. And both are females. We took away their ovaries at 4 weeks of age when they were young. And so basically there's effect of only sex chromosomes. And you can see X females died sooner. They had less survival from lupus disease compared to XY. Both are females, but XX females had worse survival. Similarly, in male mice, XX mice, these ones here, had worse survival than XY male mice. So regardless of their sex hormones, regardless of their gonads, mice that have XX chromosome have less survival from autoimmune disease than mice with XY sex chromosome. Okay. This is another test. This is a test for lupus. anyone who has anti-DNA antibody, there's a 95% chance of developing lupus. And mice that are XX, although both are females, have more anti-DNA antibody than mice that are XY females. So, repeat what I said, regardless of their gender or their congenital sex, XX mice develop more autoimmune disease than XY mice. So this leads to idea that increased prevalence of autoimmune disease in females is not because they are females, it's because of the sex chromosome doses, because they have 2X chromosome. Now, how do we take this finding and apply to humans? So this is what we started doing in 2010, and I came here to this meeting in 2011. And one approach is to take individuals with XY variation and study their immune response, or other approaches, what we call candidate XY gene approach. I go and look at each gene one by one, but the list is long and it will take me forever to study. So we came to you guys, okay, to this meeting, as well as to Eric went to Turner syndrome meeting last year in Anaheim, and we recruited individuals. Okay, and these are the number of people we have enrolled thus far: 48 XXY and 40X0, and all different combinations. And what our goal is to analyze, to study immune system components. In the next few slides, I will show you different immune cell types, immune functions, and immune-related diseases in people carrying different sex chromosome genotype. So let me talk a little bit about what is immune system, especially since this crowd may or may not know about immune system. So immune system is a network of cells, tissues, and organs that work together to defend the body against foreign invader. And these are different areas where immune system resides. And immune system fights against all kinds of invaders in the body, including bacteria, virus, fungus, cancer, or toxins. And immune system is the second largest organ in the body. Anyone who tells me the largest organ in the body, I will take that person, not you, Eric. I will take you out for lunch. I come next time.
Skin.
Skin. I didn't say the area, I say weight. It's not easy to get lunch from me. Liver. So in terms of weight. Not the surface area. Okay. All right. So immune system consists of bone marrow. That's where all immune cells are made. All the immune cells are made. The thymus, where immune cells take classes, they get educated in thymus. One type of immune cell called T cells. They go to thymus, and that's where they get their education and learn how to respond. Then in spleen, that's where the biggest what you call barrack is. That's where they come and collect, okay, and live there until they are called to action. And the lymph nodes are local offices, basically. So they are like army units in different states. So these are lymph nodes, and these are small organs everywhere in the body, all kinds of different places. And then there are other immune organs called adenoids or appendix. And these immune cells from lymph nodes or spleen, they travel in blood vessels or a network of lymphatic vessels from one lymph node to another lymph node and to different organs. And they live in intestine and they live in tonsils. These are well-defined organs, structures where immune cells live. Then immune cells are everywhere in every organ. So for example, this is a section of the skin, which one of our fellows did, and this is just the upper section of the skin, and there are two different cell types, and this is a blow up of one of the cell types. What it tells us that there's a very strong, and actually now the new technology allows us to actually image these cells in real life, and you can see these cells are constantly moving and seem like they're talking to each other, extending their arm and basically wrapping around other cells, talking to each other. So there's a very strong network of immune system. So basically what I'm telling is whatever I study is really important compared to, anyway. So the immune system, and I don't want to go into detail, the immune system is a network of many immune cells and molecules. And this is to show that this is complex and it will take me forever or anyone forever to study. Immune cells arrive from stem cells. Stem cells in the bone marrow, somewhere here, and from these stem cells, different types of immune cells arise, basically. I will drop a few words here, which will be important for my next slide, is we are really interested in cells called T lymphocytes or T cells. And there are two big groups of T cells. One is killer T cell, and one is helper T cell, and I will tell you next slide what they do. And there's a cell called B lymphocyte, which makes antibody. And there are other cells which do all kinds of different functions, and I will come to that. So lymphocytes, that is a major focus of my lab. They're small white blood cells. They play a large role in defending the body against disease. And the B cells make antibodies, and in simple terms, they attack bacteria, virus, or whatever, all kinds of different things, mostly bacteria, okay, and toxins. T cells, which will destroy infected or cancer cells, and these killer T cells kill, and helper T cells help. Helper T cells help determine which immune responses the body should make to a particular pathogen, particular bacteria or virus, and killer T cells will recognize infected cell and kill. And this is it's experiment called flow cytometry. We take your blood sample, so you know what we do with blood sample. We put in a machine, and each individual dot is a cell in your blood. And we put some colors so we can identify which cell types, and you can see individuals who have 2X's have less of this cell type. So individuals with 1X have 36, and these are 19%. We have been studying in about 30 or 40 individuals, and we see that this is statistically lower compared to people with single X. So having X might have some consequence for one cell type called CD8 positive T cell. There's another cell called CD4, and CD8 is generally a killer cell, and CD4 is generally a helper cell.
When you designate XX, are you talking about XXY, or could that be a female?
So we have done in different ways generally. So in case of this experiment, this is true for both. Okay. So XXY versus XY, or XX versus X0. So this is true for both situations. In a small number of people we have looked at, 30 or 40 total. I'm just giving a representative.
And X0 is Turner's.
Turner's, yeah, sorry. X0 is Turner's. So we compare XX versus X0 and XXY versus XY. That's how we do it. Okay. Now, so if you take these cells and give them some food in culture, what we call stimulation, then these cells proliferate, and here is a cells that is showing proliferating, and you can see the proliferation level of these CD8 cells is higher in people with single X compared to double X. So suggesting that these cells are proliferating more. They're proliferating less in people with 2X's. Now, I will warn you, this is a preliminary data, number one. Number two, this data was obtained using XXY individuals here and controls XY from our blood bank, and they were done on different days. that's why, I'm trying to, basically enroll people, both controls and XXY individuals, right at this meeting. Okay, so another important property of immune system is to remember things. So whenever someone gets infected, they mount a response, say antibody response or something called T cell response, and then the response goes away, they eliminate bacteria, but then they remember things. Okay. And when they remember things, this is called memory T cell or memory B cells. So next time you get infected, those memory cells will remember, hey, I have seen this bacteria, this virus, and I'm going to attack right away. So first time you have infection, it takes longer time to, for the immune cells to come into work. Second time, they come right away. What we find that this very simple test of just looking at the numbers of memory cells, and the cells that are not memory, we call them naive cells. So cells here are memory cells. And you can see there are differences between X and XX, 2 and 11, and this naive means the cells that have not seen, that don't have memory, they are just new cells, and you can see the difference here. There's another cell type called, yeah.
Do those cells themselves last decades, or do they pass on the memory to other cells?
There's evidence for both. So some people think some of these memory cells act like some sort of stem cells. They will be there, and then some of them will differentiate, and whenever there's infection, they will be called into work. And basically, but the pool of stem cell remains. That's what the current thinking is. So there's another cell called natural killer T cells. And I don't want you to remember all this, but basically we found that this was difference between the, so like here, this XXY, XY, because we had enough samples here, so I could put the, this graph. Basically, so their numbers were less. Now, natural killer T cells are very different T cells compared to regular T cells. In terms that these cells are ready-made, they are ready to go. They what we call innate cells. Normally T cells, whenever there's some infection, T cells need to be educated, and they take time to go to the college, and then basically eventually are ready to work. Whereas NK T cells are ready to work right away. Within hours they will work. And we found that in these cells there were differences. Okay. And then immune cells release chemicals. These chemicals are called cytokines. And these cytokines are really important whenever these cells need to talk to each other. So one cell becomes active, makes these cytokines will go and look for the cell on which it's supposed to work, and basically it stimulates or suppress or inhibit the other cell. And just so we found that between XX and X, there were differences in this cells making one cytokine called interleukin-4. We have a lot more data, but I'm not showing because of relatively preliminary nature of this data.
Just push continue.
What is that?
Just push continue.
Continue? Oh, okay.
It's just accessing.
Open hyperlink. Oh, continue. Okay, let's see. So, let's see if there's a.
Different types of bacteria. Bacteria take advantage of the cut as an entry point. When damaged skin cells come in contact with bacteria, they release chemical signals. These signals trigger inflammation at the wound site. Inflammation is a type of defense against invasion. In the bloodstream, one layer below the damaged skin and bacteria, we see a mixture of red cells, white cells, and platelets. One type of white blood bloodstream and make their way through tissues to the infection site. Eventually, neutrophils find the bacteria. On contact, a killing process known as phagocytosis begins. The neutrophil pulls the bacteria into a compartment called the phagosome. Inside the phagosome, the isolated bacteria are surrounded by swirling granules. The granules create a hostile environment by releasing killing elements into the phagosome. Protein complexes generate clouds of deadly agents to help kill the bacteria. Other granules continue to fuse with the phagosome, releasing many other different agents that also contribute to killing the bacteria. In a matter of minutes, the immediate threat to the body has been destroyed, and your immune system has successfully protected you from a cut on the skin.
So far I was discussing about lymphocytes, and I thought it would be nice to show a movie and also this group of cells, which are called phagocytes. And these cells don't need to be educated. They are ready to do their job. If there's a bacteria coming in the body, they will just go and try to eliminate the bacteria through the process you saw in the movie. In the last few months, we have begun to study these cells, and we don't have the data, but there's some preliminary indication that it would be important for us to study these cells in people with XY variations. So that the next time I come, you make sure you remember the neutrophils do this job, okay? And we'll probably have some data on neutrophils. So the third aspect is the immune diseases, different types of disease that are directly related to immune system. Immune system is probably involved in almost every disease system, but these are main ones. So one is where immune system doesn't work because these cells either not there or they don't work. This is called immune deficiency diseases. So in immune deficiency disease, when the immune system is not as strong as normal, resulting in infections. This can occur because of genes on X chromosome or on other chromosomes, or because of infections that will kill immune cells. Then there is cancer of the immune cell where immune cell becomes cancerous, like lymphomas. Then there's autoimmunity, which is kind of exactly opposite of what happens with the immune deficiency, where the immune system overacts, does more than what it's supposed to do, and it attacks normal tissue, and then asthma and allergies. These are the different types of diseases where immune system is directly involved. And we will be studying these diseases in people with XY variation over time. And that's why you have this long 10-page questionnaire. And many of you have been really upset with me about the questionnaires. So yesterday someone was saying, it's like 10 pages, it takes me half an hour. And I was lying, telling them, well, no, exactly, you can do it in 10 minutes. But actually, I was lying there. It takes time. And the reason it takes time, because in the beginning, when we started this project, we were just interested in autoimmunity, because that's what I have been studying for the last 20 years. But when we see all those immune changes, some of the early things we have found in our lab, I think we think we have an opportunity to learn more than beyond just one small area I'm interested in. And we have other people in our group who are interested in looking at infection issue, looking at asthma, allergy. So we think this is a great opportunity for us to learn and great opportunity for us to be able to contribute to this literature in XY variation. Okay. So I told you this, the main job of the immune system is to basically find something that doesn't look like belonging to the body and go and kill it or eliminate it. And how does the immune system do this job? So immune system or immune cells have to train themselves in early life. And how do you train to shoot someone? Basically find a friendly target and shoot it, right? So that's what immune cells do. They basically learn by reacting or shooting against your own body. But they do in a way that it doesn't become like what Dick Cheney did. So basically, you just make sure don't, just kill animal and not basically shoot someone. So however, and immune system has very strong ability, very keen ability to distinguish what is your own body and what is not your own body. Whenever that ability goes away, because of some defects in genes or whatever, this thing, all kinds of different things that can cause that, that's when autoimmune disease occurs. So autoimmune disease is basically, sorry for Republican side for the comment, I didn't mean to. Just every time I give this talk, it comes. So, and I showed you this slide about the autoimmune disease. There are at least 81 of them. And I showed you the people we have enrolled. And here's some of the data. So ANA or anti-nuclear antibody is a test we commonly do in our clinics in people we suspect to have an autoimmune disease such as lupus. What we find, and sometimes this anti-nuclear antibody can be present in normal healthy population. It's reason because the immune system, that's how immune system learns to shoot against itself and make some antibody. So here you can see XY individuals having some antibody, but if you look at higher titers, which are really important for disease, then you see a big difference here. So people with XXY, and this was unexpected, so XXY males have significantly higher prevalence of autoimmune disease of this test compared to XY. Okay. And so in this slide, each lane represents blood from one individual, and each lane this way represents one test. And we did about 200 different tests of different antibodies belonging to different organs in our body. So we took these, what we call antigen, something belonging to liver, a skin, a joint, and basically ran against this. And we did different types of tests called IgM or IgG, and this is a summary of that data, some of the antibody. So chromatin is an antibody that is present in people who have lupus. An anti-chromatin antibody. So anti-chromatin antibody is present in people who have lupus, and these are lupus patients, high levels of chromatin antibody. What we found that compared to XY individuals, there's significantly higher levels of anti-chromatin antibody. Not everyone. A small number of few individuals, they mounted fairly strong response. And these are different examples of different antibodies that usually present in people with lupus or other autoimmune disease. They seem to be higher in individuals who are XX males compared to XY males. And I had opportunity to talk to some of these people at the meeting because some of the samples came from some people from this conference when we were here last year or a couple of years ago. So, however, it was not like that these individuals with XY were making antibodies non-specifically with all kinds of things. There was some, for most antibodies actually there was no difference. And for some antibodies, don't worry what is coming, I'll discuss. For some antibodies, actually there was exactly opposite trend. That some of the antibodies were actually less in individuals with XXY compared to XY. So what it tells me that the XX-related factors work in different ways. It won't be such a simple story that we can say, make a blanket statement that autoimmune diseases and all these bad autoantibodies will be higher in individuals with XXY. It will be a lot more complex. The different autoantibodies will be differently regulated and different people will respond to it, which makes sense. That it's not like a blanket that one has a diagnosis of XXY and they will have increased autoantibody. That doesn't occur. Okay. This is data from X0, and I'm showing because this was kind of dramatic. In fact, this was in people with Turner syndrome. Okay, and so we had those 83 antibody tested, and Eric is here. He analyzed the data. What we found that compared to XX females, X0 females had differences in antibody. 50 out of the 83 antibodies, which was kind of dramatic. That more than half of the antibodies we tested were different between these two types of people who carry with this sex chromosome genotype. And out of what we found was more than 90% of antibodies were lower in people who had X0. What it tells us, and these IgM antibodies are made by B cells, B lymphocytes, that probably the having 1X has something to do with B. And so we're now instructed by this data, now we are going to go back to and take out samples from individuals with XXY and see if, and we'll try to find out if there's a reverse trend in people who have 2Xs, males who have 2Xs. Okay. So in summary, we have made some observations in the last 2 years in this project. We have found alterations in certain T lymphocytes and certain cytokine producing cells. And we found increased production of certain undesirable auto antibody, which normally should not be there in people who have XXY compared to XY. Okay. However, I think yesterday I discussed this, that when we looked at the questionnaire, we didn't find the people who had high levels of, actually, I was also talking to people at this meeting who had those high antibodies, whether they actually have the disease that is associated with those autoantibodies, and that doesn't seem to be the case. So either they are not telling me the full story, okay, or maybe something very interesting happening here, that they have been making these bad antibodies, but somehow their body is able to inhibit that bad response and keep the body in homeostasis or control the bad response, which is, if that is true, that would be a good news. Okay. These alterations in these different cell types I discussed might have implications for protection against infection, so we need to study. I don't have an answer for that, so that we will be studying. And these are especially important for here is that if the memory cells, actually what the preliminary observations that we have made, if they are true, then that might have implications for chronic infections. If somebody gets a fungal infection or tuberculosis or some of these bad chronic infections, then this might have implications. But we don't know this at this time. Okay. And I'm repeating this slide. So yesterday, so thank you very much. We had 34, 35 samples, so which is good. But this time is exactly reverse trend. So we have only 7 individuals with XXY, and thanks to what we call control individuals, you were really gracious to give you a sample. But if your son or daughter who have XY variations, I think it will be important because we need to be able to compare the findings. If we can't compare, I can go to blood bank and get the controls, but what I want is controls from the same family. So where we have where we have variation and the normal sex chromosome numbers, and then we can compare the findings of the two, I think that is really important for us. And as I told you yesterday, and some of these, we hope that if we continue to work hard in this area, we might have some explanations for some of these unusual findings. When I talk to individuals with XXY, many of them have some symptoms which can be explained by known disease patterns. So we hope that eventually we might be able to provide some answers. This might also be helpful in making early diagnosis for immune-related conditions. We provide vaccines for all kinds of different these infections or pathogens, and if we learn something different, then we may be able to change the vaccine, the way we give vaccine in individuals with XXY compared to XY or other people. And if this data is true, if the antibody data is true, then that makes a case for doing these screening tests for individuals who are XXY. For example, the data I showed you an anti-nuclear antibody, if that is true, if we do another set of, we study another set of individuals and we find the same pattern, then I think we can write about it and the docs can do the screening test before the actual diagnosis. There was a study done in people from military, from Army, And in Army, what they do, they take blood sample every year. And so what one of the investigators in one group actually in this town, Denver, and then another group in Oklahoma, what they did, the people who are found to have autoimmune disease like rheumatoid arthritis and lupus, they went back and tested their blood sample for 10, 15 years before the diagnosis, before they even had any symptom. And what they found was many individuals had those autoantibodies several years before they had the first pain or first rash or first problem. So we are hoping that if we make progress in this field, then we may be able to educate that that XXY individuals should get these screening tests regardless of their, whether they have symptom or not symptom. Now, once we know the test, but next question is, even if you test positive, what can you do? Right now, we don't have answers for that. Even the people who are test positive, we can't do. But there are lots of studies that are ongoing to give some treatments or even possible vaccines. They're in very early stages of the studies, which can be done in the future. Okay, and this is the website. So you don't really need these paper form. You can go to this website and have your son or daughter complete the form over several cups of coffee. and once you do that, then we will know, Eric and our team will know that you have entered in the database, and we can contact you and figure out a way to get the blood sample. We are currently struggling to get this funding to do this work. We are mostly, we had funding from the NIH to do the mouse work. Okay. And they gave a small amount of money, a seed money, to do human work. That's how we started. Right now, we're volunteering for this study. And we're hoping that when we have more data, we can obtain more funding to continue this work. Thank you. Questions? Yes.
For those of us that are here, will you take the blood sample now?
Yeah.
And also fill out the form later?
Yes.
Great.
Dr. Tartaglia had mentioned that there tends to be more respiratory infections in people with sex chromosome abnormalities. And I was wondering, has there been anything yet in your research that might point to why that is?
So if the data in a small number of people about the CD8 cells are really important for fighting viral infections.
Oh.
They're absolutely critical. And so that I discussed, and it wasn't clear, and it is not even clear to me at this time, but the first finding I showed you, the cells don't proliferate, means what it means is that normally whenever there's infection, the CD8 cells will proliferate. They become like, they expand by million times or several million times. And as soon as the infection is gone, what we call their population contracts, becomes small and goes to baseline. It seemed like, at least some preliminary work we have done, that is not happening in people with XY variation. And intriguingly, that is happening in both X0 and XXY. So that is very preliminary, but that tells that it may be just changing the homeostasis, normal homeostasis, like XXY just because this is abnormal, and X0 is abnormal, so that is happening in both individuals. So right now we don't understand why that is happening. But because there are differences in CD8 positive T cells, people can get infections and they may not be controlled easily.
One of the things, my son has XYY, and one of the things Dr. Tartaglia noticed was that a lot of the boys have asthma and also food allergies. And talking to the parents, it seems that they have, the food allergies are very specific to proteins, like soy, eggs, dairy, peanuts, those kind of things. Something on the Y chromosome,
So we don't have that data in people, but we have some interesting there's a gene on X chromosome. The name of that gene is called IL-13 receptor alpha 2. It's a long name. Okay. So basically, what this gene does, it's the product of this gene. What it does, whenever it sees a protein, a cytokine called IL-13, it just binds it and makes it ineffective. So people who have 2 X chromosomes, okay, so there's too much of that. There's a good amount of this IL-13 receptor alpha 2. What it means is the IL-13, the cytokine is being removed. Now, IL-13 works as a cytokine that can reduce certain, what we call inflammatory response, like rheumatoid arthritis type of response. So women will have less of IL-13 and can be more prone to develop rheumatoid arthritis type of thing. However, life is never simple. So in people who have just 1 X chromosome, so because that protein is less, so it's not able to remove IL-13, so there's too much of IL-13, relatively speaking. And now IL-13, there's a lot of data in animals and also in humans. The more IL-13 means more asthma, more allergy, more, also response to parasites and all kinds of things. So we don't know whether that is the explanation, but at least in mouse we have this data, and we have published the data. So, any more questions?
In avians where the heterogamic is female, do you see reversal of that pattern so that in female birds have less autoimmune disorders?
Okay.
So instead of like X and Y, so if W and Z, and WZ is female. So sort of switch from mammals. And do you see like then that whole pattern of autoimmunity risk switching?
So I should know because one of the prominent researchers, you're talking birds, right?
Yes,
Okay.
yes.
So one of the prominent researchers who works, and that's how actually I got so interested into this whole area. I don't know if I told you this story. How I started in this whole area, we have 6 minutes, so I can, 6 months, right? I have 6 minutes.
Sure.
But the story is long. So.
This gives the last sentence.
So I was not working in this area. I mean, not interested in this area. And about 7, 8 years ago, I was at my main campus at UCLA, and I had a meeting 11 to 12, and I had a meeting 1 to 2, and so I had this 1 hour, and I didn't know what to do with 1 hour. I said, well, if I go back to my lab and then come back, it will kill my half an hour. I'll just hang around. And I was walking in the hallway. I saw, and it was lunchtime. I was hungry. So I saw this. There was a conference room outside. There was the food there. I said, this says gender basis or sex basis for song behavior in birds and the vocalization in mice. Oh, it's interesting. I don't know anything about it, but I'll just sit there in that room. Okay. And plus have the food, so I took it. Sat there. Okay. It sounded a bit ridiculous why I'm sitting in the I'm a physician, I don't know why I'm sitting. But so he studied, and maybe he will have an answer to that. And so there he talked about those mice. He was just making those mice. And when I heard that, I said, oh, I think I can figure out why females get more autoimmune disease and why there are differences. So when he finished his lecture, I said, can you give me those mice? He said, no, these mice they will cost you at least $10,000 to me, because these mice were on what he was really not cared about the immune system. So these mice, what we call street mice, they have mixed backgrounds, and he was just concerned about brain development and how these mice make the female and male, how they're different mice. So we took the mice, worked for 3-4 years to modify them to become like lupus-like. So I don't have answer to your question, but I love telling this story. Every time this comes, so my greed for food led me to do this work.